This protocol is useful for determining changes in NF-kappa-B activation in cells expressing an NF-kappa-B luciferase reporter. The main advantage of this technique is it allows for a high-throughput screen of factors or conditions that lead to changes in NF-kappa-B activation.
NF-kappa-B is a transcription factor for a wide variety of cellular processes. A well-known function of NF-kappa-B is in the induction of inflammatory responses by inducing the expression of various cytokines and chemokines. Our lab is particularly interested in the induction of NF-kappa-B induced by the pathogen Salmonella Typhimurium. Here we used a HeLa cell line that is stably transfected with an NF-kappa-B luciferase reporter plasmid. Other bacteria or even viruses or chemical compounds can be used in this cell line to study the activation of NF-kappa-B. Other cell lines can also be used if you can introduce such a NF-kappa-B luciferase reporter plasmid in that cell line.
Somebody doing this for the first time may have issues properly utilizing sterile technique that is required for cell culture and bacterial work.
One day before cell stimulation, remove the growth media of HeLa 57A cells that have been grown to about 75% confluency. Wash cells with 1 milliliter of 0.05% trypsin EDTA. Replace with another milliliter of trypsin EDTA and transfer the flask to a 37 degrees Celsius incubator for 5 minutes. After the cells have been detached from the flask, suspend them in 10 milliliters of growth media. Combine 10 microliters of cell suspension with 10 microliters of trypan blue, pipette up and down to mix.